Good afternoon, everyone. Let's start the class today. Today's our topic is molecular basis of inheritance. So yesterday we had the chapter Principles of Inheritance and Variation where we learned about the genetics basis of genetics, the Mendelian experiments, Mendelian's law, and what exactly inheritance refers to, the genotype, phenotype, the variations, mutation, the chromosomal disorder, etc. So today we are going into the molecular level of inheritance. So what exactly causes the inheritance or the transmittance of characters from parent to progeny? So what is that carries these characters from a parent to progeny? And the answer is the DNA, that is a genetic material. So what is DNA? DNA is exactly nucleic acids. So nucleic acids are the macromolecules or biomolecules which are present in living cells. And Frederick Nietzsche was the first person who isolated the nucleic acids from the pus cells and he called it as nuclein. And later, Altman find out that it is acidic in nature and he named it as nucleic acids. And there are two types of nucleic acids found in living organisms. It is deoxyribonucleic acid, that is DNA, and the second one is ribonucleic acid, that is RNA. Now, moving to DNA. So DNA is a genetic material of all living organisms. Now, what do they contain? They carry coded information from one generation to another generation. They carries coded information, the information from one generation to another generation. And it is a long polymer of deoxyribonucleotides. That means it consists of so many monomeric units connected together by covalent bond. So it is a polymer actually. Deoxyribonucleic acid is a polymer, a long polymer of deoxyribonucleotides. So as we know that, for example, the proteins are also a polymer which is made up of amino acids. Similarly, carbohydrates like for example the starch, glycogen are also polysaccharides made up of monomeric units called monosaccharides. In a similar way, nucleic acids, for example, DNA consists of polyribonucleotide chain, that is deoxyribonucleotides. And the length of DNA depends on the number of nucleotide pair present in it. So that means a DNA molecule will be having nucleotides as the subunit or the monomeric unit. And in eukaryotic cell, the DNA is found in nucleus. It is a chief component of chromosomes, and DNA found in also in mitochondria and chloroplast. So we know that because of the endosymbiotic theory, according to the endosymbiotic theory, as the prokaryotes got evolved to form the mitochondria and chloroplast in the eukaryotic cells, they do have their own DNA because in prokaryotic cells, they have the DNA at the nucleoid region. They do not have a well membrane bound nucleus. They have a nucleoid region where the genetic material is present. That is the DNA. Whereas coming to eukaryotic cells, they will be having a well-bound, membrane-bound nucleus. Inside that, they will be having the genetic material, that is the DNA. Now, what exactly are the components of DNA? So actually the nucleic acids, as I mentioned, they are macromolecules or biomolecules. They consist of carbon, hydrogen, oxygen, nitrogen, and phosphorus. So the chemically DNA is composed of deoxyribose sugar, phosphate group, and nitrogenous base. So deoxyribose sugar. So this is a diagram of deoxyribose sugar. So it is a ribose sugar, in this one is a pentose sugar. It consists of 5 carbon. 1, 2, 3, 4 and 5. It's a 5 carbon sugar, that is ribose sugar. And in the second carbon they lack one oxygen. That is why it is called deoxyribose sugar. In case of RNA, they have the ribose sugar with this O at the second carbon position. They are having a hydroxyl group there. Instead of the ribose sugar in deoxyribose, they don't have a hydroxyl group. They have a hydrogen on the second carbon. So this deoxyribose sugar, then they have a phosphate group. That is, they're having PO4, that is a phosphate group. So we can see that they're having carbon, hydrogen, oxygen as well as phosphorus. And also they have a nitrogenous base. So nitrogenous base are including the pyrimidines, purines. We'll be coming into the content later. Anyway, so these are the components of DNA. Deoxyribose sugar, the phosphate group plus the nitrogenous base. So we can say that a nucleotide, what we refers to as like it is a monomeric unit of our DNA or nucleic acid. So nucleotide consists of the sugar, that is the deoxyribose sugar molecule plus the phosphate group plus the nitrogenous base or the purines or pyrimidines, we could say. So this is what exactly are the monomeric units of nucleic acid. Okay. Now, deoxyribose sugar, it is a pentose sugar having the molecular formula C5H10O4. That is a pentose sugar. And phosphate group, that is a phosphoric acid, forms the phosphate group. That is why we call it as nucleic acid. Okay, because it is having acidic in nature because of the presence of this phosphoric acid, and its molecular formula is H3PO4. Now, moving to nitrogenous base. So nitrogenous base are the nitrogen containing compounds, and these are mainly divided into two types as I mentioned, purines and pyrimidines. So what are purines? So the examples of purines are adenine and guanine. Coming to pyrimidines, it consists of cytosine, thymine and uracil. So purines are double ring heterocyclic compounds. You can see there are two rings and they are heterocyclic structural compound. It means these two structures are different. They're not similar. They're not homocyclic. They are heterocyclic structural compound. This structure is having a hexagonal structure, whereas this is a 5-ring structure, isn't it? So they're different. And the two types of purine present in the DNA are adenine and guanine. Coming to pyrimidine, they are single ring structure compound. So here you can see cytosine is having a single ring. That is a major difference between purines and pyrimidines. And the two type of pyrimidines present in DNA and cytosine, DNA are cytosine and thymine. In DNA, they are cytosine and thymine. So in the previous slide, I mentioned uracil as well. So this uracil is present in RNA, not DNA. So I just mentioned uracil as it is a pyrimidine. I mentioned it. But here in DNA, they do only have cytosine and thymine. Now we are going to learn a new term, that is nucleoside. So as I mentioned, nucleoside refers to sugar, phosphate and nitrogenous base. Now nucleoside refers to nitrogenous base plus pentose sugar without the phosphate group actually. That is, if the nitrogenous base is added, like they are forming a bond, the nitrogenous base is forming a bond with the pentose sugar molecule, and a phosphate group is not included in this particular compound molecule, then that is called nucleoside. So here the nitrogen base is attached to the C1 of the pentose sugar by a glycosidic linkage, N-glycosidic linkage. So in DNA nucleotides, the nucleosides are formed by the combination of deoxyribose sugar and nitrogenous base. So you can conclude that nucleoside is the sugar plus the nitrogenous base, whereas nucleotide is the sugar plus nitrogenous base plus the phosphate group. Okay. So actually I have only put it as PO4. It is PO4 2 minus, that is hydrophoric, the phosphoric acid. So it is a valency I have given there. Now we are moving to the naming of nucleosides. That is, this is a nitrogenous base, adenine plus the deoxyribose sugar. It is deoxyadenosine. That is the nucleoside. Guanine plus deoxyribose sugar is deoxyguanosine. It is also a nucleoside. Cytosine plus deoxyribose sugar, it is deoxycytidine, it is a nucleoside. Thymine plus deoxyribose sugar is deoxythymidine, it is also a nucleoside. Now moving to nucleotides, as I mentioned, they consist of the nitrogenous base, the sugar molecule and the phosphate group. So there are 4 types of nucleotides present in DNA. They are deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate and deoxythymidine monophosphate. You might have heard about the term ATP, adenosine triphosphate. This is an example for a nucleotide. Okay, so whereas in ATP there are 3 phosphate molecule, whereas coming to the nucleotides present in a DNA, they are having monophosphates. Okay, now, so as I mentioned, DNA is a polymer, it is a polynucleotide strand. So that means so many nucleotides are connected together to form a DNA molecule. Now, what is a bond that connect the nucleotides? It is a phosphodiester bond between the fifth and third carbon atom position of the pentose sugar. So this is the third carbon of a sugar molecule is connected to a phosphate group here, and this phosphate is connected to the fifth carbon of the pentose sugar. Okay, so this is a phosphodiester bond between the fifth and third carbon atom position of the pentose sugar. So moving to the next slide, we are moving to the double helix structure of DNA. So this was the introduction about the DNA structure actually, the DNA components. So what is DNA actually made up of? So now moving to the structure of DNA, that is double helix structure of DNA. Watson and Crick first proposed a structural model of DNA in 1953. And they also won a Nobel Prize for their work in 1962. So according to Watson and Crick model of DNA, the DNA contains 2 polynucleotide strands coiled together in a helical manner. So actually in this diagram we could see it's like given here it is a helical manner. It is like a staircase. So it is like 2 strands are there and and 2 strands and they are connected like they are connected together by hydrogen bonds. And it is exactly like a helical structure like this actually. Yeah, this will be the correct one. And it is like they are having 2 strands. Okay, and they are connected. Now, so what is the structure of DNA? Structure of DNA, as I mentioned, is a polynucleotide molecule. It consists of sugar and phosphate forms of the backbone. The base is projected to inside. The 2 strands are coiled each other and arranged anti-parallelly. That means one strand will be from 5' to 3' end and the other strand will be from 3' to 5' end. The 2 strands of DNA have a common diameter of 20 angstrom. And adenine of one strand pairs with thymine. So the adenine will be pairing with thymine and guanine will be pairing with cytosine. And adenine will be pairing with thymine by 2 hydrogen bonds and guanine will be binding with the pairing with cytosine by 3 hydrogen bonds. Now this is the structure of the DNA structure. So here you can see they are having the nitrogenous bases GTAC and they are paired with the nitrogenous bases in the second strand. They're double stranded there. They're in anti-parallel direction. That is 5' to 3' and another is 3' to 5' end where they are bonded by hydrogen bonds. Okay, and as I mentioned, the diameter is it is given here. It is by 20 angstrom. It is converted to nanometer. It is 2 nanometer, and the distance between 2 curves, that is like between a one curve and the distance between 2 curves will be like 3.4 nanometer, and the distance between 2 nitrogenous bases will be 0.34 nanometer. And this curve, the major curve is called the major groove and the small curve is called the minor groove. Curve in the sense it is like a it is whatever it is like forming like a intuition, like it is forming a curve structure when they are having a helical form of structure. Okay, so this is, they are having the sugar phosphate backbone, and they are having the nitrogenous bases also. Now moving to the next term, that is central dogma. So one-way flow of genetic information from DNA to protein is called central dogma. That means the genetic information in DNA is transferred to RNA, and from the RNA it is transferred to protein. So we know that the DNA is converted to RNA, or the information from DNA is transcribed to RNA by the process called transcription, and RNA is translated to form protein by the process called translation. So we have learned about all these processes. So we have learned about all these processes in the class. So we know actually like what transcription is, what translation is. And now moving to packaging of DNA in prokaryotes. So prokaryotes do not have a definite nucleus, we know that, and DNA is not scattered throughout the cell. It is held together with some proteins in a region called nucleoid. And the DNA in the nucleoid is organized in large loops held by proteins. Now, we are moving to eukaryotes, the packaging of DNA in eukaryotes. So in eukaryotes, the DNA is stabilized with positively charged basic proteins called histones. So in this diagram, you can see that the DNA is coiled around the histone proteins. So the histones are positively charged due to its rich in basic amino acids like lysine and arginine. So this is a question, why histones are positively charged? Because they are proteins, and proteins consist of amino acids, and they are having the basic amino acids lysine and arginine, which are having the positive charge. Now moving to DNA, we know that they are acidic in nature, they are having phosphoric acid, so they are negatively charged. So histones are organized to form a unit of 8 molecules called histone octamer. So here you can see in the diagram they are having 1, 2, 3, 4, 5, 6, 7 and 8. So histones are organized in the form of unit of 8 molecules, and this is called histone octamer. Now, this negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. So this is what exactly nucleosome is. So DNA is negatively charged. The histone proteins are positively charged. So when DNA is wound around the histone octamer, they forms nucleosome. And the nucleosome exactly looks like a beads on a string. We can see here, right? This is a beads on a string form. So this blue color, the beads are the the octamer, the histone octamers, and the string-like structure, that is the DNA molecule, is connecting all these pearl or the beads together to form like a beads on a string form. Now nucleosomes form the repeating unit of a structure in the nucleus called chromatin. Okay, so those nucleosomes are like this is one single nucleosome. So many nucleosomes are connected together to form a chromatin material. This chromatin is packed to form chromatin fibers. So here you can see this DNA is wound around the histone proteins to form, this is the chromatin material, chromatin fibers. And these are further coiled and condensed at metaphase stage. What stage? The metaphase stage, to form the chromosome. You can see they are condensed form. And now we have two terms here, euchromatin and heterochromatin. So what is euchromatin? So here you can see the diagram, the difference between euchromatin and heterochromatin. So euchromatin is in the nucleus. There are some loosely coiled regions of chromatin. They are lightly stained. That's called euchromatin. In this chromosome, you can see they are lightly stained. So this region, the DNA is loosely coiled around. They are like loosely packed. That is the euchromatin. Coming to heterochromatin, in the nucleus, more dense, the dark band, packed regions of chromatin that is stains dark are the heterochromatin. These are the heterochromatin. They are tightly packed. So euchromatin is transcriptionally active than heterochromatin. Transcriptionally active in the sense they are ready to undergo transcription, so that can be converted, and means like they can be transcribed to RNA. Now moving to next topic, That is a transforming principle. So this is an experiment conducted by the Frederick Griffith, and to show that like some molecule or some material has been transformed, which cause a change in the character in an organism. Okay. So let's look into the experiment here. So he conducted the experiment on mice and pneumonia bacteria, that is Streptococcus pneumonia. And he took two strains, that is smooth strain and a rough strain. The smooth strain bacteria produce a capsule, so it is pathogenic. That means it can cause harm to the organism. The R strain lacks the capsule, so it is not pathogenic. That means it is not harmful, it is harmless. So when a rough strain was injected to the mice, what happened? It is not pathogenic, it is non-virulent. So the mouse lives. Now, when smooth strain was injected to the mice, it is virulent, so it is harmful. The mouse dies. Now, when they injected heat-killed smooth strain, so that means the smooth strain is virulent. When it is heat-killed, it is not active. So what happens? It will not affect the mice. So what happens is that the mouse lives because the organisms are dead, because it is heat-killed smooth strain. But when they mixed the rough strain and heat-killed smooth strain, the mouse died. But exactly like what they expected was the mouse will leave, because we know that the rough strain will cause the mouse to leave, and the heat-killed smooth strain also allows the mouse to leave. But then when they mix the rough and heat-killed smooth strain, the mouse died. Why? So then they thought that yes, something has happened there, something transformed. Actually, this blue color, the rough strain was not virulent. Somebody changed them. That means the heat killed smooth strain had something, some molecule, some material inside them which converted the rough strain to smooth. Now, so what did they understood? The R strain bacteria had been transformed by the heat-killed S strain bacteria. Now, the transformation of R strain to S strain is due to transfer of genetic material. So actually the S strain had a genetic material which made it virulent, and the rough strain had the genetic they had the genetic material which made them non-virulent. Now it got transformed. That means this S transformed this R strain. So the biochemical nature of genetic material was not defined from his experiment. Then another experiment was done where they mixed protease enzyme to the heat-killed S strain and live R strain, and they find out that it transforms R strain to S strain. That means the protease enzyme is responsible for breaking down the proteins. So it was done to find out whether the genetic material was proteins or not. So if it was proteins, the protease would have degraded the proteins and the R strain wouldn't have been transformed to S strain. But it happened here. That means protein is not the genetic material. Then the next test tube, they added the RNase. That is an enzyme which cleave RNA, that is ribonucleic acid. So even when they added RNase, the R strain got transformed to S strain. But if RNA was the genetic material, the R would have been transformed to S. But if the RNA is working, when RNA is added, the RNA would have cleaved the RNA, and what will not happen? The transformation will not happen. But actually it happened here. That means RNA is not the genetic material. Now, then they added DNA. So DNA is the enzyme which cleave DNA. Now, when DNAs cleave the DNA genetic material, so what happened? It was unable to transform the R strain to S strain. Okay, that means they concluded DNA is the genetic material. Okay, now the genetic material is DNA. So actually, it is like they confirmed that the DNA is causing the transformation of R strain to S strain. But here what happened is that all the biologists were not convinced. So there comes another experiment, that is the Hershey and Chase experiment. So this is a experiment where it was proven that DNA is the genetic material. Okay, now let's look into the experiment here. So this work was done on virus that infects bacteria, and that virus is called bacteriophage. Okay, so in this experiment, you can see that this is a bacteriophage. During this information, during this like experiment, they infected the bacteriophages. That means bacteriophages will be infecting in bacteria. So what happens is that they'll be inserting their genetic material into the bacterial cell. So once they infect the bacteria, they insert the genetic material into the bacterial cell. So what happened is that what they did is that in this experiment, they radio labeled, I mean like they labeled radioactive components. That means they radio labeled a protein capsule with sulfur, that is S35, and also the radio labeled in like two different bacteriophages. They did in different bacteriophages. Here they labeled the DNA with P32, because nucleic acid, that is DNA, consists of the phosphate group. So they did the experiment with radio labeled radioactive phosphorus. And in case of the protein capsule, proteins are amino acids, and amino acids like cysteine consists of sulfur, and proteins will be having disulfide linkage as well. So they labeled it with S35, that is radioactive sulfur. Now, in the first experiment, when they added this radioactive labeled protein capsule, we know that when the bacteriophage infect the bacteria, they will be inserting the genetic material into the bacterial cell. Now, at the end of the experiment, when they measured, they find out that there is no radioactive detected inside the cell, whereas the radioactive detected in the supernatant. Okay, so that means when a bacteriophage is infecting a bacteria, its genetic material is being entered into the nucleus. That means it is not protein. By this experiment, they could conclude that it is not protein. Now, by this experiment, the second test, when they infected the bacteria which is having the radioactive phosphorus, they find out that the radioactive phosphorus is detected inside the cell. That means they concluded DNA is the genetic material. So bacteria infected with the viruses that had radioactive DNA were radioactive, no radioactivity in the supernatant. Whereas the bacteria infected with the virus that had radioactive protein were not radioactive, but the radioactivity found in the supernatant. So this means DNA is the infecting agent that made the bacteria radioactive. Hence, DNA is the genetic material, not the protein. So this is the Hershey and Chase experiment. Okay. Now, what are the criteria of genetic material? So it should be able to generate its replica, that is replication. It should be chemically and structurally stable. It should provide slow changes like mutation that is required for evolution. It should be able to express itself in the form of Mendelian character. And the protein does not fulfill the criteria, hence it is not the genetic material. So RNA and DNA fulfill this criteria. So what are the criteria? It should be able to generate its replication, undergo replication. It should be chemically and structurally stable. It should provide slow changes like mutation, and also it should be able to express itself. Okay. Now we are moving to the details of central dogma. So as I told, central dogma is a single way flow of messages or genetic material information from one organism to another. That means not from one organism to another. It is a flow of information in an organism where a particular gene get expressed. So if a DNA is copying itself to form another copy of DNA, that is replication. Okay, so it's a process by which the DNA produces the exact copies of original DNA. And eukaryotes, the DNA is double-stranded. The two strands are complementary to each other because of their base sequences, isn't it? So semi-conservative method of replication is happening there. So what is semi-conservative mode of replication? So this is an old DNA strand. They are double-stranded. So one is a template strand and another is non-template strand. Okay. So we have the new partner DNA. That means like new form, newly synthesized DNA we can see here. And in that you can see that one is the newly synthesized one and one is a old DNA strand. That is a parent strand. That is what semi-conservative replication is. So in each new DNA strand, one strand is old while other is newly formed. So Watson and Crick describe this method as semi-conservative replication. Okay, now we are moving to another experiment conducted by Nicholson and Stoll. So this experiment proved that the DNA replication is semi-conservative nature. So here what they did was they grew E. coli in heavy nitrogen medium, that is N15 medium. For example, the ammonium chloride culture medium was taken. So this N15 is the heavy isotope of nitrogen. So here we could see that both the strands of the DNA have N15 DNA. That means after generations, they found out that the DNA in the bacteria become labeled with N15, making it denser than the normal DNA. Now, after that, what they did was they shifted the bacteria to N14 medium, that is ammonium culture medium having a light nitrogen medium, that is N14 medium. So here, DNA, after that the DNA was extracted and subjected to cesium chloride density gradient centrifugations, and they find out that the DNA present in that particular medium was hybrid type of DNA, or we could say intermediate DNA. That you can see here they're having both N15 DNA as well as N14 DNA. So why it is happening like that? Because we know that the bacteria was first grown in a medium containing N15 medium, that is heavy nitrogen isotope, like heavy isotope of nitrogen was taken in the medium. So the DNA were having the heavy nitrogen. Now when it was transferred to the next medium that is having the light nitrogen, that is N14 medium, what happened? It is because of the semi-conservative replication, the parent DNA remains there, whereas the new DNA strand that is formed will be having N14. So this is the N14 one and the old one will be having N15 isotope. Sorry. Okay. So what they find? They find that there is a hybrid intermediate type of DNA. Both N15 and N14 isotopes were present. So the position of DNA bands was observed under the UV light and they find it out like this in generation 1. In generation 2, when they find it like in the next generation, they find out that they had both, like they had intermediate as well as completely N14 like DNA. They're having only N14 DNA, like DNA was present. That is after the next generation. So there is equal amount of, that means equal amount of N14 as well as equal amount of hybrid DNA were there. So from this they concluded that the DNA replication is semi-conservative. Okay, now moving to DNA replication process. So this is a process where the DNA replicates. That means they copy themselves and the substance, they need the enzymes DNA helicase, DNA polymerase 1, 2, 3, RNA primase, DNA ligase and SSB, that is single strand binding proteins. So this is the process of DNA replication. Yeah, so this is the DNA replication process. Here we have the parental DNA. This is a helicase enzyme. Then we have the RNA primase enzyme, the RNA primer, which starts the replication process. The Okazaki fragments, the DNA polymerase one, the DNA ligase, which ligate the joins the Okazaki fragments, the DNA polymerase 3, the single strand binding proteins, which keeps the DNA strands as single, that means for the replication to be done. And the strand, the newly formed strand, there are two newly formed strands, that is leading and lagging strand. A leading strand will be formed from 5' to 3', 5' to 3' direction in a continuous way. It is a continuous strand formed, whereas lagging strands are formed discontinuously, so that is called the Okazaki fragments, and they are joined by the DNA ligase enzyme. And we know that the DNA replication starts with an RNA primer, which is synthesized by the primase enzyme. So first is the initiation. So we have the origin of replication, the ORI site from where the replication starts. The helicase enzyme unwinds the DNA. The single strand binding proteins will be like holding the single strands apart, and the primase synthesizes the RNA primer. The DNA polymerase 3 is responsible for synthesizing the new DNA. Then the sliding clamps hold the DNA polymerase in the place. We have the Okazaki fragments synthesized there, which are the discontinuous newly formed DNA strand. Then by RNase H, the RNA primers are removed. By DNA polymerase 1 fills in the gaps. The DNA ligase seals the mix between the Okazaki fragments, and finally the termination. So these are the steps of DNA replication. Now, as I mentioned, central dogma is a one-way flow of information from DNA to mRNA and from mRNA to protein called the central dogma. That is the DNA transcribed to RNA, RNA translated to protein. Now we are moving to transcription. So transcription is a conversion of DNA to RNA, and the enzymes required are DNA helicase, RNA polymerase 2, ribonucleotides. So ribonucleotides, what we need is that the ribonucleotide monophosphate, which activates into ribonucleotide triphosphate by phosphorylase enzyme and phosphoric acid. Hence ATP, GTP, UTP and CTP forms. Now, the transcription begins by uncoiling. That is, the helicase enzyme will be doing the unwinding of DNA, and this starts at the promoter region of cistron. So we know what cistrons are. Cistrons are the region which are coding for a particular protein. So the RNA polymerase 2 will be recognizing and binding to the promoter sequence, and they synthesizes RNA in 5' to 3' direction. Okay, so the synthesis taking place in 5' to, this is the 5' end and this is the 3' end direction. And here, which strand has been, for which strand the complementary strand or the RNA is synthesized? That is the, so here which is this template strand? Template strand will be 5' to 3' template strand. That is, this one is the 5' to 3', it is a template strand. For that, the complementary strands will be synthesized. Now, this is the coding strand and the template strand. So for this template strand, they will be producing the RNA molecule, which will be exactly similar to the coding strand. So this is the DNA coding strand, which will be similar to the RNA molecule. Now, this is the direction of transcription. Now, as I mentioned, the complementary base for template strand pairs to transcribe mRNA, and when the RNA polymerase reaches the terminator point, what happens? The synthesis of RNA stops up. So newly synthesized mRNA in eukaryotic cell is called the heterogeneous RNA, and then they undergo post-transcriptional modification. That process called splicing. That means the introns will be removed, exons will be joined together. So the eukaryotic mRNA contains the exons and introns. In splicing, introns are removed and exons are joined in a defined order. So that is about splicing. Now, next two process happening is capping and tailing. So splicing happens, capping happens, and tailing happens. So capping is like methyl guanosine triphosphate is added to the 5' end of the hRNA. Then tailing is polyadenylate tail. Poly-A tail is added at the 3' end. Now the processed hnRNA is called the mRNA, which is ready for translation once it is transported out of the nucleus. So this is the mRNA, messenger RNA, and it carries the genetic information in the form of triplet codons. So they are having this 5' cap and poly-A tail, and this is the coding region. A start codon is here, a stop codon is here. So we are moving fast because we have to go to the questions. That's why. Okay. Now we are moving the next type of RNA, that is rRNA, that is ribosomal RNA, which helps in the binding of ribosomes to mRNA. So this is the we know that ribosomes are the protein synthesizing machinery. So ribosome synthesizes, or they carry out the process of translation. And ribosome consists of RNA, and that is called the ribosomal RNA. So this acts as an enzyme as well, that is ribozymes, which helps in the formation of peptide bond during protein synthesis. So what are the difference between DNA and RNA? DNA are high molecular weight, whereas RNA has very low molecular weight. They are double-stranded. DNA are double-stranded. RNA is single-stranded. DNA ribosugar is the deoxyribosugar. In RNA, it is ribosugar. Now coming to nitrogenous base, they have thymine instead of uracil, and RNA has uracil instead of thymine. Now in case of DNA, unusual nitrogenous bases are absent, whereas in RNA, they have unusual nitrogenous bases. Now coming to the DNA, they are the genetic material of the living organism, and in case of RNA, they are the genetic material of some viruses. Now, we are not moving to the concept of gene right now because we have already learned it in the last class. So we know the terms like cistron, as I mentioned, is a structural gene. It is a functional unit of DNA that codes for a specific protein. Then recon, it is a unit of DNA that undergoes recombination. So reco is referring to the recombination part. Okay. Then muton, it is referring to the term mutation, that is DNA that capable of undergoing mutation. Now moving to replicon, it is a unit of DNA that undergoes replication. Now genetic code, it is a 3 specific nucleotide sequence that code for specific amino acid present in DNA. For example, A, U, G, it is a start codon which codes for the amino acid methionine. So this is a genetic code. Now, this is a single codon, a 3 specific nucleotide that codes for a specific amino acid in mRNA. And this codon will be like DNA will be having multiple codons. For example, AUG, CAG. It is not, sorry, it is, I'm drawing mRNA as I am having the U here. So AUG, CAG, AAT, CUT, like that. Okay, they are made up of codons. Now, what is anticodon? So, the 3 specific nucleotide sequence code for specific amino acid present in the anticodon arm of tRNA is called the anticodon. So, we know that tRNA will be having a triplet codon in them, which is specific to particular amino acid, which will be complementary to the codons. For example, if AUG is the codon in mRNA, the tRNA will be having UAC as the anticodon. So this is the codon and UAC is the anticodon. Okay. Now, what are the general features of genetic codon? The genetic code is triplet. They are universal. There are no overlapping is there. They are comma-less. They are degenerative in nature, and they are somewhat universal as well. So these are the different genetic codons, and they will be coding for a specific amino acid as well. Now we know the initiator codon, the start codon. We have the stop codon, that is OAG, UGA, and UAA. And genetic code is unambiguous, that is each codon codes for a specific amino acid and only one amino acid. Okay, and it is unidirectional. The codon reads only in 5' to 3' end. And next is the Boole base, that is in each codon, third nitrogenous base is less specific. Okay. Then this is the structure of tRNA. As I told, they are having the anticodon at this particular area, that is anticodon arm and loop. Then they have D arm and loop. Here they have the T5C arm and loop. And this is the acceptor arm where they carry the amino acid. So this is the translation process. This is the mRNA molecule which binds to the ribosome, which is having two subunits, the small and the large subunit. So this mRNA will be read by the ribosomes, and they will be like having three different sites, A, P, and E, where the tRNA molecules will be carrying the particular amino acids along with the anticodons. So this anticodons will be complementary base pairing with the codons present in the mRNA. At the same time, what will happen? They will be having a bond with the amino acids, which is carried by the consequent tRNA molecule. So if the first tRNA is carrying methionine amino acid, it will be at the P side, and the second tRNA will be moving to the A side carrying the second amino acid, and then they will be having a peptide bond linkage, and finally they will be synthesizing a complete protein molecule. Okay. And at the end, when they reaches, as you can see here, they're having the start codon as well as stop codon. This is the stop codon here, UAA. So when they reaches the stop codon, the process terminates, and finally the synthesized protein is ready. That is a polypeptide. Now moving to operon. So operon is a group of genes that are transcribed at the same time. It is a sequential arrangement of regulatory gene, promoter gene, operator gene, and structural gene prokaryotes, which is called the operon concept. And it is consisting of three structural genes just involved in processing the sugar lactose. And one of them is the gene for enzyme beta galactosidase. And this enzyme hydrolyzes lactose into glucose and galactose. So what happens is that when glucose is present and lactose is absent, the E. coli does not produce beta galactosidase. And when glucose is absent and lactose is present, the E. coli produces beta galactosidase. Now coming to Human Genome Project. So what is this at? Like the government started the project in 1990, which coordinated the Department of Energy and the National Institute of Health. So here, the whole hereditary information of an organism that is encoded in the DNA is called genome. And human genome have approximately 3 into 10 raised to 9 base pairs. So what they planned is to identify the approximate 20,000 to 25,000 genes in the human DNA, and to determine the sequences of the 3 billion bases that make up the human DNA, and to store this information in databases. And they also plan to improve the tools for data analysis and to address the ethical, legal, and social issues that arise from genome research. Now moving to DNA fingerprinting. So we will be like taking sample from a particular organism, for example, a plant leaf, and the extracted DNA will be cleaved by the restriction enzyme, and they'll be then fragmented by electrophoresis. Then we'll be like undergoing the Sagan blotting, and that means we'll be using a probe, their radioactive probe, which will be binding to the particular DNA fragment. And finally, it will be viewed under an X-ray film, and we can compare the DNA molecule and find, for example, if we want to compare non-DNA and a sample, for example, parenting issues and all, we can use this technique to find out like whether the DNA sample matches with the control or the sample to be studied. Okay. So this is DNA fingerprinting technology was used to identify the individual at genetic level, and it was developed by Alex Jeffries in 1985. And one more thing is very important. This involves identifying differences in some specific regions in DNA, which is called the repetitive DNA. So they'll be looking for the repetitive DNAs. Okay. So yeah, we have to conclude the text here because we have to go to the questions as we are having limited time. We'll be like discussing the remaining topics in the next class. We'll go for some questions over here. So yeah, this is from September 2021, botany, question number 5. The question is DNA strands on a gel stained with ethidium bromide when viewed under UV radiation appears as what? Is it bright blue, yellow, bright orange, or dark red? The correct answer is option C, because after staining, the band appears bright orange under UV light. With staining, it is ethidium bromide staining. It is an intercalating agent which binds between the DNA bases. So it is a direct easy question. Now moving to the next question, which is the following is a correct sequence of steps in PCR. So this is a polymerized chain reaction. We haven't discussed in our class right now. So this is a process where we amplify the DNA. Okay, so that means if we are having a sample of DNA and we want to proliferate it, we want to multiply, we want to amplify, we want to make so many copies of DNA, we use the process called PCR. Okay, it is simply replication happening here. So the first thing what happens is that is a denaturation. Okay, that means annealing and extension is incorrect. Now once the DNA get denatured, they add the primase. That means that the RNA primer, the primer and will be added to it. That is that process called annealing. Okay, so that means annealing is the second one, correct? Now once annealing is done, the primer is added to this means it annealed to the DNA strand, they extend. That means they'll be adding the nucleotides, nitrogenous base, sorry, not nucleotides, nitrogenous bases, adenine, guanine, thymine, cytosine, like that. So extension will be happening. That is the third process. So it starts with denaturation, then annealing and extension. So option B is the correct answer. Now moving to next question from September 2021, question number 21 from botany. That is, during the purification process for recombinant DNA technology, addition of chilled ethanol precipitates out what? So it is exactly the DNA. That is recombinant DNA technology. We use it for like creating our own gene of interest, not creating and inserting it into a host cell and making it more copies. So we want, for that we need to isolate the DNA first, right? So isolation in the sense we have to separate the DNA molecule from all other molecules. So we will be having polysaccharides, RNA, histones from that. So we can use enzymes like protease and RNase to relieve or remove the proteins and RNA. And now we have to precipitate the DNA. And we use the chilled ethanol at the end where we can extract the DNA molecule. Okay, so the answer is option C, DNA. Now this is the central topic. Okay, this is like what we learn right now. We know, right? It is from DNA to mRNA to the proteins. So D will be the proteins, and DNA to mRNA is called transcription, and mRNA to protein is called translation. So here D will be the correct answer. A is so yeah, A is here. So I missed it. A is replication. That is DNA is like copying themselves is replication. So B is transcription, C is translation, and D is a protein molecule. It is easy question, direct question. Now, this is from September 2021. This is again related to PCR. So this is question number 29 from botany. So which of the following is not applicable of PCR? Okay, so PCR is used to detect the gene mutation. This is true regarding PCR. For molecular diagnosis, for gene amplification, we use it. It is related to DNA. But option D is incorrect. Purification of isolated protein. This is nothing related to protein. PCR related to the isolation of DNA, not proteins. That was also an easy question. Okay, now moving to the next question. What is the role of RNA polymerase III in the process of transcription in eukaryotes? So here it is about the transcription process, and RNA polymerase I transcribes the 5-S, 18S and 28S RNA. That means they'll be transcribing, in the sense they'll be producing these rRNAs like 28S, 18S, and 5-point-S rRNAs. And coming to tRNA polymerase 2, they transcribe the hnRNA. Okay, that is a precursor of mRNA. So that is a precursor of mRNA. So this is done by RNA polymerase 2, and this is done by RNA polymerase 1. Now here we have remaining snRNA, only snRNA, and here we have tNRNAs, 5S rRNA, and snRNA. So RNA polymerase 3 transcribes like all the tRNAs, 5S rRNAs, snRNA also. So that means option A will be incorrect. So option C is the correct answer. Okay. Now it is about DNA fingerprinting. So it involves identifying differences in some specific regions in DNA sequence called as what? So as we like now we learned about the topic, and the answer is repetitive DNA. So DNA fingerprinting, they look for the difference in specific regions of DNA, and that is the repetitive DNA. So the basis of DNA fingerprinting is the VNTR. It is also known as satellite DNA. There is repetitive DNA probe showing a high polymorphism. It will be different in different individuals. Whereas coming to closely related individuals, it will be similar, somewhat similar. So in case of a criminal case and all, if you have sample of the place of crime or the near to the victim, you have some sample of the culprit, and you have a person who you doubt, like you are having a person you think he is the person behind that crime, and you have a sample of that particular person's blood or any kind of sample. So you can look for the DNA fingerprinting and find out whether they are having similar repetitive DNA, that is VNTR similar or not, by doing DNA fingerprinting. Now moving to the next question, that is question from September 2021, question number 46. So you have to find out the correct statement from this. So first question is, the split gene arrangement is characteristics of prokaryotes. This is incorrect. Actually, we learned it right now. It is a characteristics of eukaryotes, because the post-transcriptional modifications is happening in eukaryotes, the splicing, capping, polyethylene, everything. So introns, exons are present in eukaryotes, not prokaryotes. Option B, in capping, the methyl guanosine triphosphate is added to the 3' end. This is incorrect. We know, right? It is in the 5' end. Okay. So option B is also incorrect. Option C, RNA polymerase bind with Rho factor to terminate the process of transcription in bacteria. This is true, isn't it? So Rho factor is actually associated with the termination process. So they alter the specificity of RNA polymerase to terminate the process. So option C is the correct answer. Option D, the coding strand in a transcription unit is copied to mRNA. This is incorrect. Actually, it is not the coding strand. It is the template strand which is being transcribed to the mRNA. The coding strand is similar to mRNA. This is a non-template RNA actually. Coding strand is a non-template RNA. We use the template strand to copy that will be copied to the mRNA. So option D is also incorrect. As you can see here, this is a template strand of DNA which is being they will be complementary to the mRNA, whereas a non-template strand RNA will be the coding strand. Now, moving to next question, DNA polymorphism forms the basis of what? So here the answer is option B, both genetic mapping and DNA fingerprinting. Just now we mentioned that DNA polymorphism, in the sense every individuals are different, because they have different the genome. Though they are having similar genes, the sequence will be different. Okay, so gene mapping of the human genome and DNA fingerprinting rely on the polymorphisms in DNA sequence. If you take your population also, they will be having different the repetitive sequence, the VNTR, the SNRs, everything will be different. So we can use this to identify an individual from a population. As I mentioned in the criminal case, as I discussed in case of DNA fingerprinting, it is similar here. There is DNA polymorphism. Okay. Now let's move to the last question. So in this question, it is given that in an E. coli strain, the I gene gets mutated and its product cannot bind the inducer molecule. So if the growth medium is provided with lactose, what will be the outcome? So we know that if lactose molecule, the inducer is present, what will they do? They will bind to the repressor protein. And repressor protein is coded by the I gene. And as the I gene got mutated, they will not be able to bind to lactose even if it is present in the medium. So if repressor is not able to bind to the lactose, what will happen? The repressor is able to bind to the operator, and which will stop the expression of ZYA genes. That means the ZYA genes will not be able to transcribe or translate into the particular proteins. So option B is the correct answer. So we are winding up the class right now because of the limitation of time. We'll be continuing the chapter, the continuation of chapters as well as questions in the next class.